things regarding plant protection, and also we would like to share with each other about our experiences with in plant protection, and hopefully with this guest lecture activity, this can broaden our knowledge. Also, through this activity, we would like to see the opportunity to strengthen our partnership or relationship with other institutions, or even maybe we can build a new collaboration with other institutions. So before we start this very first guest lecture, I would like to explain the rule for this guest lecture. Yang pertama, Mbak Ana mungkin bisa ditampilkan slide informasi. Yang pertama, para peserta dimohon untuk menonaktifkan speakernya selama guest lecture menyampaikan kuliahnya. Jadi mohon semua bisa tertib ya. Kecuali nanti di akhir presentasi moderator memberi kesempatan untuk bertanya secara langsung. Tapi kalaupun tidak, mahasiswa atau Bapak dan Ibu dosen boleh menuliskan pertanyaannya di kolom chat pada Zoom pada saat presentasi oleh guest lecture kita. Jadi siapa tahu pas menyampaikan sesuatu terus teringat ada sesuatu yang mau ditanyakan, itu silahkan langsung dituliskan di chat room ya, supaya nanti bisa dicatat oleh panitia. Kemudian ada link kehadiran bagi mahasiswa atau Bapak Ibu yang memerlukan sertifikat, nanti akan disiapkan oleh panitia. Baik, saya kira itu. Oke, Bapak dan Ibu, dalam beberapa menit kita akan memulai kuliah yang akan disampaikan oleh Pak Stephen. But before starting that, again, I think it's a good time to take group pictures since everybody is here. Siapa yang bisa bantu, Bu Lia? So please open up your video if you would like to be included in the picture. Oke, Bu Lia, ya, bisa berhitung.
Ya. Oke, sebentar.
Ada berapa ini?
Ada 6, Bu.
6 ya.
Ya, sebentar ya. Saya mulai 1, 2, 3. Oke, sebentar. Kemudian untuk yang kedua, saya mulai 1, 2, 3. Kemudian ketiga. Ya, 1, 2, 3. Halaman berikutnya. Oke, 1, 2, 3. Ya. Oke. Baik, yang terakhir. Ya, sudah, Bu.
Okay, thank you, Bu Lia. All right, so we are starting now, and I would like to welcome Pak Widodo as our moderator for this guest lecture. So please, Pak Widodo, you may start the lecture.
Okay, thank you, Basti, and good morning, all audiences and Dr. Stephen Harper. I hope you are all fine today. This morning, I'm very excited to be a moderator of our guest lecturer, Dr. Stephen Harper. And this morning also, we are so honored and get a very good chance to have the opportunity to listen lecture from our first and very valuable guest lecturer, Dr. Stephen Harper. Dr. Harper, today's lecture has been attended by about 130 audiences. They are from the fourth year of undergraduate, and then also some graduate student, and some are alumni from Department of Plant Protection. Before the lecture started, I would like to introduce our guest lecturer today. His name is Dr. Stephen Harper. He was graduated from Queensland University for the BS, MS, and PhD in 1988, 1993, and 2005 respectively. Before joining the University of Queensland, Dr. Stephen Harper is active in as a senior principal research scientist in Department of Agriculture and Fisheries Australia. Just last month.
That's correct.
His position is last month. Yes. His position is an associate professor at the University of Queensland, and he was involved in many projects. I said, and you can see in the presentation, but two from three projects are in Indonesia. I think were in Indonesia. Okay, in Indonesia as crop health and nutrient management of salads and any other horticulture crops. And his specialist now as precision nutrition and soil management in vegetable production in relationship with sustainable production improvement. Without further ado, I would like to you to join me in welcoming of Dr. Stephen Harper. Please, Dr. Stephen Harper, it's your time for presenting your lecture. Thank you.
Terima kasih, Pak Widodo. I will now share my screen with you and start my presentation. Okay, sorry, can you, I'll just have to wait for you to exit your screen share, Pak Widodo.
Pardon?
Stop.
Can you
Stop.
exit screen share from your computer?
Yeah. Anna, stop. Yeah.
Oh, perfect. And then I will share my, I'll just start, hang on. Okay. Right, and then I will try and share my screen with you. Hang on. Sorry, Asti, I'll just have to shift my screen around, sorry. No, it's okay. I've just got to shift my screens around so that everything will work. Okay. You're right. No problem. Okay. Right.
Rafi mungkin apa?
Yep.
Bisa
okay.
bantu? Okay, actually. Is it okay, Dr. Harper?
Yep.
Or you have to change what is the setting? Well, okay, that's okay.
You've got it?
Okay, that's okay. Yeah, that's all right.
Yeah.
Good.
So I will just go through my presentation with you. I can't see my notes that were here, so I won't worry about it, but that's okay. I'll just go through the presentation. So as you've I've been introduced. I'm an associate professor with the University of Queensland. I've had 30 years experience in vegetable production systems. My undergraduate training was in plant nutrition and soils, and my master's was in acidic soils. And then my PhD was in physiology of vegetable cropping. So I have a relatively broad understanding of agricultural systems, and I've spent quite a substantial amount of field work diagnosing problems. And so the presentation that I will make today will not be lots and lots of data. In fact, I don't know that I've got much data at all, but it will be very much a visual presentation and an accumulation of images that I've collected from field problems that I've seen over the years. It is not a comprehensive set of disorders in vegetable crops. I stress that now. It is just a selection. And so as young scientists and extension officers, you need to understand that there's a very broad number of disorders that can affect plants, and many of them. I think I'd just like to start by giving you what I consider to be a key message. And that key message is for young agricultural scientists is that experience, careful observation in the field, and accurate diagnostics are always required in diagnosing any field disorders that you might see. Different disorders can have the same symptoms. And so I've underlined there, as you can see, accurate diagnostics is always a key part of understanding what the problems are. And I think particularly when you're looking at more from the pathology perspective and the plant virology sections, that the diagnostics are really important, and of course they're evolving all the time. So the presentation, I'll go through an introduction of who I am and why that's actually important to know where I come from, because one of the key things about disorders is that they are very much environmentally influenced. And so what you can experience in Indonesia is very, very different from what I would experience in Australia. And then I'll go through a series of the disorders in terms of a limited number of bacteria, viruses, herbicide effects, physiological disorders. Then I'll go through nutritional disorders, and then I'll actually bring them all together to talk about, well, how can these symptoms all be confounded with each other? So when I say confounding, I mean what appears to be the same thing is actually different, meaning they are confounded. We don't actually know. And then finally, I'll just go through just only 2 field case studies that might be of interest to you. I will start by apologising for my Australian accent, and I hope that I speak slowly enough that you can try and understand what I'm saying. So if at any stage you think I'm speaking too quickly, please let me know. I do understand how difficult it can be to learn someone else's language, and I admire you all for putting up with me speaking in English. Having spent 10 years researching in Indonesia, you would think I would know a lot more Bahasa than I do. I only know sedikit. So please put up with me as I proceed. So in terms of who I am and where I come from, obviously a map of Australia. And within the map of Australia, you can see the great state of Queensland, which is where I come from. And particularly in the southeast part, which I'm hoping that you can see my mouse arrow. Is that appearing for you? You can see that, great. I come from the southeast part of Queensland, and in particular, this area, name here is the Lockyer Valley, but it's actually only about that far from Brisbane, which is about 80 kilometres west of our main capital city of Queensland, which is Brisbane, and that's about 80 kilometres. And we're at a latitude, sorry, it's not showing, we're at a latitude of about 27.5 degrees south of the equator. And of course, you in Indonesia are probably about 5 degrees off the equator. So being in Indonesia, you are in the true wet tropics, whereas where I am in Queensland, are the true subtropics. And so our weather patterns that we get in my part of the world are very, very different from the weather patterns that you have in your part of the world, where you have a well-defined wet season and a well-defined dry season. In my part of the world, our weather patterns are much longer duration. So we tend to have, firstly, I'll talk about our patterns of weather shortly, but just to show you where we are, I said that we're in the Lockyer Valley, west of Brisbane, and it was nice to hear Asti say about collaboration. And so I'm now part of the University of Queensland's Gatton campus. There are two large campuses, one at St. Lucia in Brisbane, and then the campus of the Gatton facility, which is based in the Lockyer Valley. Excuse me, sorry. I thought I had that on silent. And so I'm based at this campus here, and for the last 25 years, up until last month, I was at this facility here. So I've moved across the road. The facility is about 1,000 hectares of land at the Gatton campus, and it is a campus that is developing greatly. In terms of, I talked about the climate that we have here. I've got a nice photo of a dust storm during a drought. On the left, it looks like it's raining. That is actually dust. So we tend to have extended periods of dry. And when I say extended periods of dry, it is 8 years since we have had a major flow in our river systems here. So whereas you have annual major flows, it is now 8 years since we've had a flow. Then when we get rain, we get a lot of rain. And so our systems are very diverse. There's no set patterns for our weather events. So this really impacts on the types of problems that you can experience in the field, and why you always need to consider the weather patterns and climate that you're working in when you're considering the types of problems that you can get. And I'll start with the bacterial diseases and how these can be confounded with other types of symptoms. And this particular one here is Xanthomonas campestris, which is a black rot in brassicas. And you can see there that one of the main symptoms that you get is a chlorosis on the margin of the leaf usually. It will extend into the main part of the leaf, but usually along the margin. These start as basically necrotic lesions, and the lesions are usually associated with the hydothodes. The hydothodes are pores, like openings on the external margin of the leaf where guttation of water from the plant under pressure is released. And so because these are pores in the leaf margin, they're sites where the bacteria can come in. So you can see there that whenever there is a major hydothode, you can get an infection of bacterial diseases such as Xanthomonas campestris. Diseases like this one are usually a seed-borne disease. Now, I'm going to use the word usually a lot, because although we can have general ideas on how a pathogen or a nutrient disorder should perform, I'll state this now, there are always exceptions to the rule. So although we have a general belief on how things will work, it doesn't always follow the rule. So for things like Xanthomonas, it is both a soil-borne pathogen and a seed-borne pathogen. But in general, when we have this problem, it is a seed-borne problem. Other bacterial pathogens can create quite different symptoms. So this is another pathogen that's bacterial, Erwinia Caritavora. And depending on the crop species, it will present as different symptoms. They are a pectolytic bacteria, meaning they break down cell tissue and pectins in particular. And so in a crop like lettuce, so this is an iceberg lettuce, You can see there that we have Erwinia Caritavora. The disorder is known as jelly butt because we call this, the cut surface is the butt of the lettuce, the bottom of the lettuce. And so it's called jelly butt because the central part of the stem turns to jelly because of the influence of the bacteria. It essentially only affects the parenchyma tissue of the central part of the stem. You can see there that it's not affecting the rest of the lettuce. It's not affecting the root system, but it causes severe breakdown of the tissue, the parenchyma tissue within the stem of the lettuce. So although it doesn't actually affect the eating quality of the lettuce, it is a real problem for a farmer because visually, our markets are visual type markets, it's not acceptable. This is quite an interesting one because you will hear from a lot of people that this is a problem because of too much nitrogen, or some people will say that it's a boron deficiency, both of which may give you symptoms like that, or boron particularly can, but in actual fact, it is just simply the pathogen causing the problem. And under the appropriate conditions, which are usually wet conditions where soil is washed onto the surface of the lettuce, if you can imagine, if you can look at my pointer there would normally be wrapper leaves. So these are the wrapper leaves, and this head would normally have wrapper leaves. When the soil is washed down the wrapper leaf, you can see there that the site of entry of the pathogen is actually that little spot there. So the pathogen can wash down, it enters into the tissue and then breaks down all the tissue, and so you end up with jelly butt. But again, the disorder, it is a pathogen not related to boron deficiency, and I will show you boron later in potato. The Xanthomonas species are a particular problem because they are widely distributed across a lot of species. So crops like rice and tomato and chilies, peppers, there are a large range of Xanthomonas species. And of course, the diagnostics of pathovars are improving greatly as we are developing PCR techniques and sequencing. In this particular example, it's the same species as Xanthomonas campestris in brassicas, but it's a different pathovar. So in this one, it's vitians, which is the lettuce form, and it produces slightly different symptoms. This is a particularly bad case of it, but you can see on those older leaves that the infection has actually started along the edge of the margin. So you can see the worst part is actually this V formation here, and the pathogen has then spread from that position. The important thing that you notice with the bacterial infections in lettuce is that it forms what we call a black greasy spot. So if you think of grease, as in the oil, the lubricant, it produces what look to be shiny lesions that look what we call greasy. And so that's a typical symptom of vitians in lettuce. This is a further bacterial pathogen in bunching onions. And this was a first detection for Australia that myself and one of our colleagues worked on to identify this. And I'll actually talk about this one a little bit later because I'll go through the process as a case study of how we actually diagnosed this one because it was quite a complex pathogen. But this is Pseudomonas syringae, and it was the first detection in Australia for alliums. I'll just say there, in this situation, it is actually producing symptoms that aren't typical of a bacteria. So we're seeing yellowing, which is not normally something that we would say is a bacterial pathogen symptom. I'll move now on to the viruses and talk about what viruses look like. In general, viruses produce symptoms that can be similar to other disorders, and in particular, nutrition. In the example of the image that I've shown on the left, we've actually got some garlic plots where we've got poor seed. So poor seed means a small seed. Garlic, similar to bawang merah, is vegetatively propagated mostly, garlic is only vegetatively propagated. And so the problem with viruses is that from season to season, there can be a build-up in the titer of virus. So unlike many other viruses that you get in other crop species, in garlic, you can have uniform infection, and therefore the problem doesn't necessarily look like it's a virus because it's uniform, and you would normally think that nutrition causes those sorts of problems. But in this situation, it is a virus problem. The symptoms, when you have severe virus infection, are like these symptoms here, where you have what we call chlorotic lesions. And in fact, these are so chlorotic that they're actually white. And so these lesions appear on the pseudostem of the garlic. And they're a very, very good indicator of high virus load in your garlic. So that is one of the symptoms of viruses. That symptom that you see there on the right is not a symptom that you would associate with other disorders such as nutrition. It is very much a virus symptom. This is a begamovirus in eggplant, and it's a relatively common one that I've observed throughout Southeast Asia. One of the things that defines it as a virus from a visual perspective, as opposed to nutritional ones, is that the chlorosis is something that would be consistent with both virus and nutrition. However, in the case of this begamovirus, all of the leaves are affected. So older leaves through to younger leaves are all affected by the virus. So you can tell in this situation that it's a virus because nutrition will not cause symptoms for that type of symptom over the whole plant. We would only expect to see that sort of symptom on a nutritional basis on the very youngest leaves and not uniformly over the plant. I think Ibu Asti was showing me a slide the other day of a weed with viruses, and I think you've now profiled a lot of these viruses. I think it was the same weed, Ibu Asti, and weeds are often a very good indicator of some of the viruses as well. And in this situation, I'd encourage you to probably look at, when you're looking at field diagnostics, always consider not just the crop you're looking at, but also the other plants, weeds that are in the field as well, because they can be an indicator for other problems, or trying to pick out whether it's nutrition or whether it's a virus. So when you see viruses in weed species in and around your crop species, you may think that those viruses could translate into crop viruses as well. The symptoms, again, you can see that on this particular weed, it has vein clearing. So the veins are white, putty or yellow, and therefore it's not a symptom that we would normally associate with crop nutrition, which would be the type of disorder that you would compare it with. This is a particular problem in Indonesia, and it's pepper yellow leaf curl virus. At a simple glance at the crop, you could say, well, it looks like it could be nutritional. However, in the field, you would see that it can be sporadic through to high infection, and the symptoms tend to be uniform over the whole plant. It's not in focus at the base here, but you can see that the symptoms are uniform right down the whole plant. I should have picked out a better one. It's not to say that there's not an influence of plant nutrition involved in this, and there has been work done on the leaf curl viruses, the begamoviruses, to look at trace element nutrition and its relationship with the expression of the virus. And there is actually some good information to show that things like high manganese, low boron, low zinc can make the symptoms of some of the viruses worse. So, but in this situation, the problem is a virus. The cucurbits are a particular group of family of plant that have a large number of viruses. And in fact, probably some of the vegetable species that most have viruses would include the solanaceous group of plants, which is your potato, chilies, tomatoes, eggplant, the cucurbits, which includes pumpkins, squashes and gourds, and also the alliums, which of course includes the onions, the garlics. Those groups of plants tend to have very high incidence of a large number of viruses. And so the cucurbits have quite a large number of viruses. Just out of interest, to show you a comparison of what a virus would look like and nutrition would look like, on the left there is a sweet potato leaf with feathery mottle virus. And one of the key symptoms of that is the veins are a little bit yellow. So you can see there that for about 2 to 3 millimetre width, they're yellow, but the interveinal area remains relatively green. In contrast to that, and the leaf on the right is actually zinc deficiency. You can see there that with the nutrient disorders, the veins actually remain green and the interveinal area is yellow. So these are actually almost a contrast of each other. And that delineates really one difference of some of the viruses with nutritional problems. Herbicides are always problematic in vegetable production. When I say problematic, they can cause problems very, very easily through farmer mismanagement of those chemicals. If you think about the activity of herbicides, some of the new herbicides are active at rates of only 300 to 400 millilitres per hectare. So when you think about how intense the effect of herbicides can be on crop species and weed species, it's not surprising that a major problem can occur very easily through mismanagement of them. In this particular situation here, you can see there that this is a mild exposure of glyphosate on potato and on lettuce. And the typical symptom of these is that it's a bleaching, so the green tissue becomes white or yellow, and there's pretty much a very obviously very poor growth subsequent to that. The interesting thing is that in this particular case, it's mostly the tissue that gets exposed to the glyphosate that is affected. So this tissue here remains green because it didn't get a dose, but the tissue here is expressing the symptoms. The symptoms of Roundup or glyphosate from a nutrition perspective mimic something like iron deficiency. And the reason why sometimes viruses, crop nutrition, and herbicides can all produce very, very similar symptoms is that all of them can impact on components of plant biochemistry that produce these symptoms. So for example, in the example we have here for glyphosate, the action of glyphosate is on some of the iron-containing chemicals of the plant, and therefore, because it's affecting those components of biochemistry, it can produce symptoms that are similar to iron deficiency when they're at very low concentrations. So in this situation here, if you didn't know it was glyphosate, you could say, well, gee, it almost looks like it's got low iron, but of course we know it's not. So some of the symptoms, this explains why some of the symptoms of different disorders can appear to be the same. Other herbicides, such as particularly the hormonal herbicides, like the 2,4-Ds and the amides, they produce a range of symptoms. In this particular example, this is spray drift of 2,4-D over cucurbits. And you can see there that the leaf margin has curled up and the leaf itself is folded. And so one of the key things about these herbicides is you don't necessarily get necrosis. It doesn't kill the tissue, but it affects the hormonal balance in the plant, and therefore it expresses as these types of symptoms. Another example of a hormonal herbicide in potato is there. In this situation, you can see that the growth is all distorted. You can see that there's multiple veins produced along that leaf. And with a lot of the hormonal type symptoms, when you've had spray drift or it's in the seed itself, the symptoms keep developing as the plants grow because they're highly mobile in the plant. So you can see here that some of the older tissue can recover, but the hormonal herbicide is continually relocated to the growing point and expresses itself there. So in general, these types of herbicides are expressed on the youngest growth. For the hormonal herbicide damage, they really don't look like most other disorders, with perhaps the exception of maybe mite damage, or some of the viruses can give you symptoms similar to this. But they do tend to be quite unique. The other group of herbicides that can produce symptoms that look similar to other disorders are ones like the triazonone ones. And in this one, this is a product called Sencor. I think it's Metellachlor. And it's used in potatoes a lot. And under certain conditions, you can get accumulation of the herbicide within the farming system. So it's mobile in sands, and therefore it can concentrate in the furrows and therefore express itself on crops like potato. The symptoms that you get here are different from some of the virus. You can see here that it's vein clearing. So the veins are going yellow. It's quite a wide width of vein that is yellow. And the other tissue, the main part of the leaf, remains green. The difference between this and, say, viruses, which I showed you earlier with vein clearing, is that the symptoms are not necessarily expressed over the whole leaf. So you can see here that it's on this part of the leaf, but it's not on that part of the leaf. On this one, it's only on one leaflet, and then the rest of it doesn't have any symptom at all. So in contrast, you would generally find that the viruses would be expressing themselves more uniformly in all of the veins. And of course, I may touch on it later on, gathering the farmers' records on what they have done is an important step in understanding what the problem is that they might have in their crop. Now, I'll just briefly talk about some of the physiological disorders. The physiological disorders are a little bit unique in that they do tend to be more regionally specific in whether you would see them or not. So for example, under the conditions that you have in Indonesia, you would have a very different set of physiological disorders compared with what we would have in my part of the world in the subtropics. I didn't talk about it earlier, but in my region, we have a very good environment for growing a range of crops. So we can grow temperate vegetables, so things like lettuce and broccoli, cabbage, potato. We can grow them in what we call our winter months. And in our summer months, we can grow tropical vegetables like sweet corn, green beans, pumpkins, tomato, capsicums, and so forth. So in our winter production system, we would grow a lot of lettuce. My region probably grows 30 to 40% of Australia's lettuce. It has a very, very good environment for it. But one of the disorders that we get because we're growing in the middle of winter is we get sun damage in the middle of winter. So the red dotted section there shows you that this lettuce leaf around the head of the lettuce is actually got a yellow patch on it, okay, a region of yellow tissue on it. If we look at the rest of the lettuce leaf, it's actually green. The cause of this particular disorder is because when you get excessive sunlight in winter, but the temperature is very low, the plant can't deal or use the energy that it is receiving. And so as a response to the energy that it's receiving when the temperature is low, it breaks down its chlorophyll to use up the energy that it's receiving. So instead of being able to grow, it will go a yellow color because the chlorophyll is reduced through sun damage. In areas where it's not exposed to the sun, it is a normal green color. So you can get physiological disorders based on the temperature, by sunlight, by weather conditions. These disorders here are a physiological disorder in potatoes. And I showed you earlier the one of lettuce with Erwinia Caritavora. This one's a different disorder. So this one occurs under a range of conditions. It can happen through chemical damage as well. But in this situation, it's a growth-related disorder. And it causes a central breakdown in the tissue of the tuber. And if the conditions are what we call anoxic, meaning there is no oxygen in the soil or it's a very wet soil, then the tissue actually goes a black color. So depending on the conditions that you're growing your crop under, you can either have a mild hollow heart or a severe hollow heart. Hollow means a cavity in the tuber. A third type of physiological disorder that we get at our latitude again. So again, I stress knowledge about your own climate is really important. And this particular disorder is a photooxidant damage. We get it in Australia, and you can see there, it's where the tissue is exposed to the atmosphere, particularly when there are inversion layers of air currents. So high pressure air conditions coming through the country, through Australia, create conditions that result in the production of ozone, and the ozone burns the leaf tissue. So in this diagram, you can see that this image, you can see there parts are really badly affected and some parts are not affected. Similarly here. And the difference is where the tissue is not directly exposed to the atmosphere, that is, if it's got some leaf cover over it, it doesn't show the symptoms. And so you can see here on this fully exposed leaf and leaves here, there is expression of the damage from the photooxidants, but in the tissues that are protected and lower, so if you look down in there, they don't express the symptom. So it's not a pathological one. So if you go and do an isolate on that, you will not find a pathogen. It does not get worse with time. It just stays exactly the same. So it's not a pathogen and it's not a nutritional disorder. It is just simply a problem that you get because of the environmental conditions that you're growing the plants under. The physiological disorders are often the more difficult ones to understand because we don't have good diagnostic tools as we do for nutrition and for some of the pathogens or most of the pathogens. So some of the physiological disorders can be very difficult to understand. Now I'm going to move on to nutritional disorders. And I'm not sure how much understanding everybody has of plant nutrition, but I'll very briefly give you one slide about plant nutrition. We basically divide, as scientists, we basically divide plant nutrients into the macronutrients and the micronutrients. All of the macronutrients and micronutrients, when we run the diagnostics, are expressed on the plant dry matter basis, okay? So we dry the tissue and then we analyze it using a range of techniques and come up with a value. For the macronutrients, which consists of nitrogen, potassium, calcium, phosphorus, sulfur, magnesium, we express them as a percentage of the dry weight. So for a 3 to 4% dry matter for nitrogen, that would mean for every kilogram of dried plant tissue, there would be 30 to 40 grams of nitrogen. If it was 100 grams, it would be 3 grams. So we express them on a percentage. I have actually highlighted here these in order of crop uptake. And you will notice that nitrogen and potassium are the most dominant nutrients taken up by plants. Calcium comes in at third at about 1%, and phosphorus, sulfur, and magnesium are all about the same at about somewhere in the order of 0.2 to 0.4%. These are just rough figures here that I've put in here, but around about that. The interesting one, of course, is phosphorus, because in all of your studies, I'm sure you hear about NPK. Why a lot of scientists talk about N, P, and K, it's not about the plant's uptake of phosphorus that puts it in those top three. It's more of a soil function. So in volcanic red soils, and particularly those of Southeast Asia, phosphorus fixation is a major problem that reduces the availability of phosphorus. So it's not a reflection, talking about NPK is not a reflection of the plant's demand for phosphorus. It is a reflection of the soil's availability of phosphorus. So the actual uptake of phosphorus by a plant and its requirement for phosphorus is substantially lower than what it is for N and P and calcium. When we look at the micronutrients, again, we express them on a dry matter basis, but we talk, instead of terms of percentage, we use milligrams per kilogram, or if you wish, you could use ppm. If we converted the value for iron at 400 milligrams per kilogram, it comes out at 0.04%. So it's substantially lower than the macronutrients, and that is the definition of the micronutrients, that they're required in lower amounts. So in order of requirement, you can see there that iron is about 400, manganese 150. These are rough figures. You will find that there's a very broad spread of these concentrations across plant species. Zinc, boron, copper, molybdenum. Now, the important thing to understand is that just because molybdenum is 0.5 milligram per kilogram in the plant tissue does not mean it's any less important than nitrogen. All it means is that the plant needs more nitrogen than molybdenum. If a plant is molybdenum deficient, it will not matter how much nitrogen or phosphorus or potassium you apply. Molybdenum deficiency will be the key determinant of how well the plant grows. So all of them are equally important in their role in the plant. It's just that the uptake is different. So when we look at nitrogen deficiency, the defining feature of nitrogen deficiency is that it expresses itself on the oldest leaf. And I'm sorry for this image, it's not the best quality image, but you can see there that on the oldest leaf, it is a real yellow color. So nitrogen deficiency tends to produce a uniform chlorosis, yellowing, of the oldest leaf. So you can see in the young leaves, it's actually broccoli, you can see in the young leaves that these are really quite green still. If we look at a field picture of nitrogen deficiency, you can see here that it can actually be quite patchy. But what you will, patchy meaning there's a yellow bit there where it's more yellow, but it's generally yellow all the way through. So you can see nitrogen deficiency, and I'll use the marker to show you nitrogen, 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 nitrogen. So you can see that it's uniformly affecting the whole crop, but it can actually be worse in some parts of the farmer's field. So it's worse, it's worse in this patch here because that part of the field is lower. So because it's lower and it gets wetter, it loses more nitrogen from the soil, and you see more nitrogen deficiency in that spot there. On the right, you can see a close-up of how these plants look, and you can see that it's a uniform yellowing, and then the tissue goes what we call necrotic, it dies, but it still just looks a yellowish color. A typical way that nitrogen develops is on the oldest leaf, and this is a better photo, and you can see on both Chinese cabbage and on broccoli, the oldest leaf is showing the symptom, and it usually starts as a V, in a V-like pattern. So it started there, and then it progressively builds out as a V. And the same if we look over here on this leaf. It started there and it progressively goes and goes. This is generally one of the symptoms that would set it apart from Xanthomonas that I showed you earlier, where the Xanthomonas, apart from that lesion there, which is still part of the nitrogen, Xanthomonas would form lesions all the way along the margin, whereas for nitrogen deficiency, it usually comes out as a V. The other symptom that you get with nitrogen deficiency would be a purple colour associated with low nitrogen status. When you see purpling on these sorts of leaves, it usually reflects that the chlorophyll, the green pigment, is reduced, and what you're seeing is the red pigment that was a background colour. So when you lose the green, you can see the purple, and that makes the difference, depending on the species. So again, sometimes with nitrogen deficiency, you will see a purpling of the foliage, and particularly if conditions are cold. So for this symptom, in most parts of Indonesia, you may not see that symptom simply because you have hotter conditions than we do in Australia. So there is an interaction between the temperature that the crop experiences and the expression of nutritional disorders. We move on to phosphorus, and it's somewhat similar to nitrogen, and in the sense that the oldest leaves are affected. So if you look at this large older leaves, you can see it's yellow first, but then it goes what we call necrotic. So the tissue dies. So it's yellow and very quickly goes to a brown to black color. It's not a gradual change. It suddenly goes necrotic. You do not see the symptoms. You do not see the symptoms on the youngest leaves. So you can see there that the youngest leaves are healthy, and it's the older leaves that are affected. The one disease that you could probably confuse this with would be verticillium wilt, which is common in the eggplant. But in this situation, verticillium wilt would normally just affect one side of the plant, which is why it's called verticillium, whereas other half of the plant is actually healthy. So the fact that it affects uniformly the older leaves, so you can see older leaf, older leaf, older leaf, older leaf, young leaves are good, it's more likely a phosphorus deficiency, and in this case it is. If we look at magnesium, magnesium of all of the mineral disorders is probably the only one that I comfortably can diagnose by my eye in the field. Magnesium deficiency, and these are mostly ornamental plants, produces strong yellow or white interveinal chlorosis, and the veins remain very strongly green. And it's a wide green section. So that's about 6 to 7 millimeters of green tissue. And the margin between the yellow tissue and the green tissue is really very well defined. It's not a gradual change. It suddenly changes from green to yellow. So they are quite distinctive. When we look at capsicum, it is one of the crops that I have found in my travels that mostly expresses magnesium deficiency. We've actually been doing research on the magnesium uptake of capsicum and found that there appears to be genetic control of magnesium uptake, which is not well documented. So what we find is that some of our chili varieties that are wild chilies have far better magnesium uptake than does the commercial cultivar, which shows strong expression of magnesium deficiency. So an important thing is, and it doesn't just apply to magnesium, there's a broad genetic difference both from species to species and within species in terms of the expression of nutrient disorders and the demand of individual species for particular nutrients. So we find that crops, for example, such as the alliums, have a very strong uptake for sulfur and a very low uptake for potassium, whereas crops like potato, for example, have a very high uptake of potassium and a relatively low uptake of sulfur. So there is no one recipe that works for all crop species. They are all different. Zinc deficiency is quite a prominent deficiency in almost all farming systems. In sweet corn, which this is a picture of, it is a species that is particularly prone to expressing zinc deficiency. It has a very high demand for zinc compared with other crop species. In sweet corn, the symptoms express themselves as interveinal chlorosis. So remember, in sweet corn, it's in the grass family, so it's a monocot, a narrow-leaf plant, and so the veins run linear and parallel. So you can see there the parallel veins, they remain green, and the interveinal area, which is that little section through there and through there, goes yellow. One of the defining features of zinc deficiency in sweet corn is that you usually just get 1 or 2 sections where all of the veins go yellow. So it appears as a broad section of yellow tissue in a linear direction. These are just examples of zinc deficiency in other crop species, including capsicum and sweet potato. The thing with zinc deficiency is that it will express itself in the youngest tissue. So for most of the micronutrients, and particularly zinc, copper, manganese, iron, and boron, they will express in the youngest foliage, not the old foliage. Whereas the macronutrients, most of them express themselves in the older leaves first. There are exceptions, so I make that clear. Zinc deficiency will often occur in farms where soil has been moved and you expose subsoil. So in Australian farming systems, we would level ground using a tractor, and often ground is removed from one part and put over in another part. So in those parts of the farm where the soil is cut down to a subsoil, This often expresses zinc deficiency. The mechanisms we're not sure of, but the expression is really quite strong. So you can see here a strong expression of zinc deficiency where they have moved soil from that farm to level the farm. Boron deficiency is quite a common deficiency in acidic soils and in alkaline soils. And unlike most nutrient disorders, it expresses itself in a more random way. So in a field that's affected by boron deficiency, we can have one seedling really badly affected. We can have one seedling intermediately affected and one that might not be badly affected at all. And as this particular crop approached maturity, you can see the severe boron deficiency, and adjacent to it is a plant that's not expressing boron deficiency. The reason for this is likely, firstly, that the seedlings that the farmer purchased were low in boron, and when these get planted out in the field, the ones that are a little bit lower don't develop a good root system to capture boron, and so they don't grow particularly well. So you end up with a situation with boron deficiency where you can have plants that look okay, but plants that look severely deficient in boron. The symptoms of boron deficiency are probably not easily confused with most other disorders. They don't really look like virus symptoms, and they don't really look like other pathogens, so they are quite unique. With the exception of probably mite damage that produces a toxin. So mites can produce toxins that can give symptoms similar to boron, and I'll show you an example of that later. So you can see here that one of the symptoms of boron is, you can see the oldest leaf on this seedling had grown well, but a total cessation, a ceasing, a stopping of the growth at the growing point of the plant. The other thing is that the leaves are often very thick and they feel like leather. So like skin, hard skin, they feel very, very thick. And this is due to, when you have boron deficiency, there is an accumulation of starch in the leaf because the plant can't biochemically process sugars properly and transport sugars. And so the plant tends to accumulate starch within the chloroplasts of the leaf cells, making them very, very hard and leathery like starch, like leather. So when something is very boron deficiency, if you try and fold the leaf, it usually breaks because it's actually brittle. It's hard and it just cracks in half. This slide over here on the right is also a boron deficiency, and it does look very similar to PYLCV. So in some situations, these deficiencies can look like some of the viruses, and in this case, it does. If we were to take a microscopic view of the lettuce that I showed earlier, and you go and look at it at about 50 to 100 times magnification, you can see that one of the symptoms of boron deficiency is the cessation of the growing point. So the growing point is what we call blind. It's not active. It's not going to produce new leaves or new buds. And the other part of the symptom that you get with boron deficiency is the production of polyphenols, because the metabolism of the plant to produce the phenolic compounds that a plant needs is disturbed. And so the plant produces brown melanin pigments. So it's just brown tissue that's not healthy. If you were to try and isolate a pathogen, there is no pathogen there. It is just simply a physiological problem. So nutrition is a physiological problem, but they are a specific type of nutritional problem for which we can often diagnose them a lot better. So the physiological browning is associated with the boron deficiency. Manganese deficiency expresses itself on the youngest leaves. So this is a potato leaf, which is a compound leaf. So this is actually a leaflet. It produces very fine green veins. So in contrast to the magnesium that had really wide green ones, these are very fine, very small. And the interveinal area is quite yellow. And the margin between the green and the yellow is not a strong division. It's a gradual change from green to yellow. And the symptoms of manganese can be a little bit variable in crop species. So we have a pepper, capsicum, chili on the left, and on the right we have a tomato. Both of these are manganese deficiency, but expressing themselves slightly differently. So you can still see the greenness in there, but in severe manganese deficiency, it may well become a yellow colour. Iron deficiency looks very, very similar to manganese deficiency. And this again is in a weed species. But the difference is that the veins are green, so it's parallel veins, it's a monocot plant here. But the tissue in between the veins tends to be more of a white colour than it is a yellow colour. Now, one of the things about iron and manganese as elements is that they both sit next to each other in the periodic table. So manganese has an atomic number of 25, iron has a number of 26. And so from a reactivity perspective, they are very, very similar. And so in plant nutrition and plant biochemistry, manganese and iron can, for some reactions, partially replace each other, meaning some of them have common reactions where they can be active. And so because of the similarity in terms of their chemical attributes, they produce symptoms that are at least similar to each other. So if you look at the previous one, you can see fine green veins and yellow. And then when we look at this one, fine green veins and yellow. So the symptoms of manganese and iron can be really quite similar. And for nutritional deficiencies, the final one that I'll just talk about is molybdenum. And molybdenum is the element that's proven to be essential that is required in the lowest amounts. So the plant uptake of molybdenum gives you a concentration of about 0.5, I'll say, parts per million, which is a very low concentration. But having said that, a deficiency of molybdenum is a serious issue. So although it's required in very low amounts, its effect on crop growth is still substantial. This is a photo of molybdenum in cucurbits, and there are well-defined genetic differences in plant demands or plant species demands for molybdenum. Of all of the elements, it's probably one of the strongest ones for the micronutrients. So for the cucurbits, which are pumpkins and squash, zucchinis, the demand for molybdenum tends to be high relative to other crop species. So the symptoms of molybdenum deficiency are really quite varied. There are not necessarily a single set of symptoms. There are a range of symptoms produced depending on the species. So in cucurbits, it will produce a general yellowing, not dissimilar to virus infection. And in general, it will often affect the older leaves first, causing a yellowing and then subsequent necrosis. It looks very similar to nitrogen deficiency because molybdenum is a cofactor in nitrate reductase, which within the plant is the process by which it's converting nitrates into ammonium and into protein and chlorophylls and other proteins. And so when you have a deficiency in molybdenum, it can look like nitrogen deficiency because it's such an important cofactor in the conversion of nitrate. However, unlike nitrogen deficiency, and this image won't quite show it as well as I'd like, on the older leaf, I showed you earlier that nitrogen deficiency causes chlorosis. In molybdenum deficiency, it will also cause chlorosis. However, you also get necrosis because there is an accumulation of nitrate to toxic levels that the plant can't use, and it causes the oldest leaf, which you can just see there, to actually go necrotic and die. In contrast to what you see in cucurbits, in brassicas, molybdenum is also often a major deficiency. In particular, on acidic red soils, molybdenum can be a major problem. And you can see in this particular example, this is a disorder called whiptail, W-H-I-P-T-A-I-L, whiptail. And this disorder affects the expansion of the crop leaf, the plant's leaf blade, and it doesn't expand properly. And so the symptoms of whiptail are there. This is a fairly specific symptom in brassicas, so broccoli, cabbage, cauliflower, that have relatively high molybdenum demand. I'll just finally talk about a few of the disorders that we get that are more generic or general type problems. And in Indonesia, on your upland acidic soils, and in Philippines and most parts of Southeast Asia and Australia, problems of aluminium toxicity or what we call acidic soil infertility are often a problem. These do not present symptoms that are well defined. They can be a range of symptoms. But I've included here just some images of some of the types of symptoms. So there can be purpling, there can be stunting, it can go in this picture here of coffee or cocoa, I can't remember. They look yellow and there's necrosis, but it produces a range of symptoms simply because there's a whole range of problems that go with acidic soils. Salinity is another problem that causes necrosis, usually on the oldest leaves. And the photos here I've just shown you where there's large patches of salt in this particular soil that cause symptoms of salinity, is another problem. So confounding of symptoms, and I'm just going to quickly go through some of the symptoms that we have. Confounding can occur where you've got multiple nutrition disorders, there's a lot of field variability, and different disorders that produce the same symptoms. So for example, if we have multiple nutrient disorders, on the left, we have a combination of zinc and boron deficiency. On the right, we have a combination of zinc and iron deficiency. And so when you get combinations of deficiencies, plants look a lot sicker, and the symptoms are not what you might find in the textbook. So the symptoms can be varied when you've got multiple disorders. When we look at the field variability, it's often a really good indicator as to whether the broad type of problem that you've got. In general, if you look at this photo on the right, if you see that sort of symptom, you would be very sure that it's a virus problem, because it's not a uniform effect in one part of the paddock. There is no pattern to how it's expressing itself. So you can be fairly sure that that's a virus problem. However, sometimes field variability can occur with nutrient disorders. And so I mentioned about boron earlier. On rare occasions, there can be times when nutrient disorders can give you a lot of field variability. So a small plant, a medium plant, and a large plant is indicating a high level of variability. Field variability, and I mentioned about garlic earlier, because it's clonal propagation, vegetative propagation using cloves or bulbs, then you can get a uniformity associated with poor seed that is related to virus infection. So if you had a whole farmer field with plants that look like that, you could be mistaken by thinking that it's a nutritional disorder, when in actual fact, it's the planting material itself that was so uniformly infected with viruses that it gave a uniform crop that was poor. So the symptoms in this situation are actually reflecting a virus problem and not nutrition. The symptoms of some of different disorders, for example, like I talked about vein clearing earlier, so yellow veins in each of these, but one is a virus, one is actually urea, fertilizer toxicity, and the third one is a herbicide. All of them are giving you yellow or clear veins with interveinal green, but they are all of a different cause. Similarly, in a way, I've got the virus and urea toxicity on the left, where there's vein clearing and green interveinal, and it's almost the opposite when we look at manganese and iron deficiency, whereas in this one, the veins are green and the interveinal area is yellow. In this one, the veins are yellow and the interveinal area is green. So they're almost like a photographic negative of each other. So I'll quickly just go through, if we've got time. Do we, Pak Widodo, do we have time?
Okay, I think it's okay.
It's okay?
Yeah, okay, it's okay.
Yeah, good. So I'm just going to go through 2 case studies and about how we went through a process of diagnosing one, a pathogen problem, and the second one, a nutrition problem. So the first one was the disease that I talked about earlier, which was Pseudomonas syringae, which was a problem and a new disease for onion and shallot in Australia. When we looked at the fields, the farmer phoned me and said, I've got a problem with my bunching onions. Can you come and have a look? So we went and had a look. And the interesting thing was we had chlorotic, so yellow, patches of plants throughout the field. So down there, there's some over there, some there, some over there, some there, some there. And the interesting thing was that they were mostly on this side, stronger on this side of the irrigation than they were on this side. So there's a little bit there, but they were prominent where the sprinklers were. So where the water comes from is there and there and there. So you can see there and then there and then there. These were associated with the irrigation system. So it made us think that it really shouldn't be a virus and it shouldn't be nutritional. So our thoughts would have been that it would be something fungal or pathogen related, but the evidence of the yellowing was not something that we would have associated with either a bacteria or a fungi. And so our preliminary guesses were, well, perhaps we are actually dealing with a virus because the yellowing and being not uniform, but in patches. So it's not the whole planting that's affected, it's just patches, made us think that it's more likely a pathogen, not nutrition. We would have expected nutrition to generally affect the whole crop. So we went through diagnostics of PCR and ELISAs to identify whether we had a potential virus. These all came back negative. I spoke with our pathologist and said, can you run a PCR for a phytoplasma? They ran the PCR for phytoplasma. It came back negative. So at that point, we'd eliminated the viruses, we'd eliminated phytoplasma, which really left us with nutrition as an easy one to identify, or something more difficult to understand. We didn't think it was a fungal pathogen. So because it was easy, and even though we didn't believe it was the problem, we ran a nutritional diagnosis on the plant tissues, and we had the yellow ones, the yellow shallots. Now, in Australia, we call a bunching onion a shallot, so please excuse me. So a yellow shallot or bunching onion, and a green one, which is the healthy one. The only thing that we could find that was really different in the analytical profile was that nitrate nitrogen was substantially greater in the yellow shallots compared with the green ones. When we look at all of the other analytes, so phosphorus, potassium, sulfur, calcium, nitrogen, sodium, chloride, they were all actually higher in the sick shallots than they were in the healthy ones. So the sum of all of that information indicates to us that we are unlikely to be dealing with a nutritional problem. So as a research scientist, my first thought was, I must have missed something, that I got something wrong, or I haven't seen something that was important in the field and I've missed it. So I subsequently went out late on a Friday afternoon when I should have been finished work, and I had another look at the field, and one thing stuck out to me. The symptom that we saw as the yellow symptom was not the problem. So the youngest leaf, the very youngest leaf was yellow, the newest leaf was yellow, but the leaf below the yellow leaf was necrotic on all of the tips. So for every yellow leaf, the leaf that was older, the leaf that was below, the yellow leaf was the one that had the infection in it. So I took samples of this particular piece of leaf here that was not the yellow one, took it back to the laboratory, cut it, put it in water, and it oozed. So the oozing from the cut plant tissue indicated we were dealing with a bacteria. So on that basis, I then phoned our pathologist and said, I think we're dealing with a bacterial pathogen. So because we didn't have this pathogen in Australia, I wasn't expecting to see a new pathogen. So my advice would be always be prepared to expect the unexpected sometimes. So we went through a whole series of complex diagnostics to identify, which took around about 18 months to achieve, to identify that this was a new Pseudomonas syringae pathovar in onions in Australia. So again, it was really about going through the appropriate diagnostics, being vigilant and looking carefully in the field for all of the symptoms that are present. The final one that I'll go through as a case study is improving a farmer's crop nutrient management program. And I've just shown an image there. It's a piece of an image, so please excuse it. But you can see in this particular image that it is patchy. There's some green plants through there. There's some yellow ones there. There's some green ones there. There's some tip death there. The problem presented itself as being a general yellowing of the crop that was not as healthy as it could be. So with that farmer, we then went and did a plant tissue test on his crop. And the thing that stood out was that for a bunching onion, the nitrogen was a little bit low. We like to see that value there at around about 4, not 3. The potassium we also like to see at about 4 and not 3. And the boron we like to see at about 30, not 11. And the zinc we like to see at about 35 to 40, not 19. Now, rather than deal with a lot of nutrients at the one time, I would also add that the manganese was also low. But farmers use mancozeb as a fungicide, and mancozeb contains quite a bit of manganese. So in his spray schedule, he would be applying manganese as mancozeb. So we developed a program to improve nutrient management and compare it with the farmer's own practice. And so we set up an experiment where we looked at the farmer practice plus foliar zinc, which is by foliage, by spray, and boron. The only reason that treatment was in there was because the farmer wanted to put some on. He never put zinc or boron on previously, but he didn't want to not put it on. So I couldn't get a treatment where there was no trace element. The farmer didn't want to do that. He wanted to have some on, even though he never did that. So when we look at the farmer's practice plus foliar nutrient, we had a fresh yield of 23 tonne per hectare. If he applied soil zinc but didn't increase his nitrogen and potassium, he got the same yield. So this indicates that since there was no, so this is a farmer practice which is low nitrogen, low potassium, low nitrogen, low potassium. So when there was not enough nitrogen and potassium supplied, there was no yield difference. However, when we went to our recommended practice, which was a twice the rate of nitrogen, twice the rate of potassium compared with his rate, we got a substantial yield improvement. And in particular, once we got the N and K up to a high level, we got a further effect of adding zinc and boron in the soil. So our yield improvement from the farmer's practice, even though he's used foliar zinc and boron, was about 7 tonne per hectare. So about a 30% yield improvement. And importantly for bunching onions, it gave a quality improvement because product is sold on the basis of having very nice blue-green foliage. And if it's not blue-green foliage, it doesn't get a good price. So by improving crop nutrition, the quality of his product and yield were both improved. So in that field, you can see this is our best practice in the background there. So you can see that that's a real blue-green colour. And in this part is the farmer's standard practice. So we were able to demonstrate to the farmer that by addressing nutrient constraints, he could improve his yield and his quality. So just my final points for key points for vegetable field diagnosis. And this is not a full list, okay? Not a full list. There will be more things you can think of. But some of the ones that I think of, when you go into the field, have an open mind. If you're a plant pathologist, don't just think it could be plant pathology. If you're a plant nutritionist, don't just think it could be crop nutrition. Have a broad, open mind that it could be a range of things. Observe your plant symptoms and the field conditions very carefully to take into account the soil profile, irrigation, all sorts of things in the area, weeds and things. Look at all of that. You need to talk to the farmer themselves, okay? So review the farmer's practices. Find out what the farmer's actually done. What did he use as fertilizer? What chemical sprays did he use? What herbicides did he use? So also talk with experts. Go and talk to people who have knowledge about pathology, agronomy, fertilizer, soils, and also look at references. And I guess the other thing that's probably very important is to understand the climate, the crop itself, and the soil that you're working with. And then finally, sometimes you have to expect what is the unexpected. So sometimes new diseases, new pathogens, new problems emerge that you hadn't expected to be there. It doesn't often happen, but it does happen. So it's important to do that. So finally, terima kasih. These are my cattle, my sapi. So
Okay.
thank you very much, and I'll stop sharing my screen and allow
Yeah.
you to just talk.
Okay. Thank you, Dr. Harper. I forgot to inform you that some of our audiences here are also the faculty member of Department of Plant Protection. One of them are graduated from Queensland University, Pak Joko.
Oh, very good.
Okay, and then it's a nice presentation. Now I just take the last good point is from the last slide, that we have to be an open mind and then expect the unexpected. I think it's a very good, to be a good diagnostician. And then diagnostic is very important and it's a critical point for us to solve the plant health problem. The accurate, carefully observation and something like that is very important to make a strategic solving for the plant health problem. And one also I wrote here, one important thing that for a long time, mostly in our farmers in Indonesia, we always said NPK. I mean nitrogen, phosphorus, and kalium. But now it should be changed with NCK. Okay, so
Well,
it's a good point. And then I think many farmers in Indonesia and ignoring the micronutrients.
yes.
And this is also sometimes it's very critical, either in directly disorders, nutrition disorders in plant, but also in the resistance of plant to some disease.
I think almost certainly, yes.
Yeah. There are so many questions in chat, for example, from Pak Gianto and from Mr. Arif. But actually, it is as already answered in your last what is the last slides. What is the main clue or the basic step in diagnostic for all of our expert. We are all from plant protection. So sometimes we just mind with the what is the pathogen, but maybe forgot what is the other disorder or the other symptoms. So there's maybe you can explain more about what is the last slide, that is maybe the answer for Pak Joko and Pak Gianto about the step diagnostic, the good diagnostic in the plant health problem. Please, Dr. Harper.
I think the thing too is that I probably haven't highlighted is that I work in vegetable production, and vegetable farming systems are far more complex, I believe, than are other cereal-based systems or pulse systems or other systems that are less complicated. So, you know, the vegetable crop species have a large range of pathogens, a large range of including bacterial, fungal, and viral. They have a large number of insect pests, which are directly related to some of the vectors for viruses. And they also have a lot of inputs of fertilizer and irrigation. And so they tend to be systems where you are more likely to get a lot of problems as well. So I probably just should say that. I think the thing that's most important is to know people who are very good in their discipline. So, for example, having worked with Ibu Asti for a long time, I know she's a very, very good virologist. So when you have problems, think broadly, but do get the right advice from people who are good diagnosticians within their discipline. That's really, really important. In my career, I've always had very, very good plant pathologists to work with. And that's why I've sort of learnt a little bit about plant pathology, and it's certainly been extremely helpful for me as a vegetable agronomist.
Okay, thank you. There's any questions, but maybe it's also the same with related with diagnostic tab. Are there any possibilities using the what is like image processing or what kind of like a photograph in for the first step of diagnostic?
Sorry?
I mean using image processing. No, it's like biosensor and something like that. Is there any possibilities to the first step for diagnostic?
Theoretically, yes. There is a group here at the University of Queensland that's been looking at specific sensors for image analysis, but I think we're a long way from doing that.
Yeah.
The problem, I think, is that if we just looked at nutrition, for example, in a field situation, if you fly a drone over, it will all give a yellowish reflectance pattern. And you probably can't really say, oh, well, this is the problem. The other problem, if we look at, say, nutrition, we don't normally see the worst case symptoms. And usually what we get, if I can actually show you, if we look at, I'll just find one slide and I'll share that with you. Just be with me. Okay. Oops. So if we look at, I'll just share my screen with you. If we look, can you see that? Oh, sorry, hang on. Can you see that screen now?
Yeah, okay.
Yep.
Yeah.
If you look at that one on the right, it's actually a really, it's not a really bad deficiency of something. We sometimes use the term in plant nutrition called hidden hunger. And hidden hunger means a crop that's hungry, but you can't see it. So it is a little bit deficient, but it's not so bad that you'd say, wow, I've got a real problem here. So when some of the nutrients are at just slightly lower than optimal, it's often actually hard to see that that's there. So developing sensors to pick up a hidden hunger is really, really difficult because they're not at optimal, but they're not obviously deficient either. And so I think that the whole issue around developing sensors is great. I think application of molecular and gene expression probably will give us far greater sensitivity in understanding the impacts of these in the future, is my belief. So, yeah. Did that answer the question?
Yeah, I think so. Yeah. Thank you. And then there's another question about what is the herbicide toxicity.
Yeah.
Okay. It's from Mr. Prabowo, the student of undergraduate students.
Yes.
Regarding the herbicide effect and symptoms that produce on the main crop. Based on your experience, what kind of vegetable that most susceptible are then and tolerant to the herbicide effect, and how long?
For which effect?
I mean, what kind of vegetable that most susceptible or tolerant to the herbicide effect, and how long you say that symptoms disappear? It can be this
herbicide.
Yeah, herbicide.
Yeah. Yeah. My short answer is I don't know.
Okay.
Herbicides are very, very difficult, and the margin for error on using them properly is very narrow. So when you have problems with herbicides, they're a big problem. So usually plants don't recover particularly well when they've had a little bit of herbicide damage. They're one of the more difficult things to deal with because they're active at such low concentrations. When you put it over a hectare, you know, you've got 1 litre over the whole of a hectare and it can kill plants. So the tolerance and the margin for error for getting it wrong is very, very small. You know, they are a problem.
Okay. Thank you. And then that's the next question from Prabhavati, from Ms. Prabhavati.
Yes.
Hi, Stephen. Thank you for your sharings. How to determine, especially to farmers, which methods are suitable for farmers' best practice? In your case earlier, you suggest to apply the microelement boron and zinc through soil application than foliar. Is it dependent on the mineral that we want to apply, or something like that, or another one?
Yes, it absolutely depends on. It actually depends on your soil.
Yeah.
So in some soil types, you will get a better response out of foliar application. One of my PhD students working on garlic in solution culture seems to think that foliar application is better than putting it in through the soil. So I think it's an area we don't really know real well. And it depends on your own farming system as to which works best. So for myself, when I run experiments now, everything is on drip irrigation. So I always put all of my nutrients through the drip irrigation because they're put right at the root zone. So in Indonesian farming systems, it will be different. And you also have to identify whether you have a need for the micronutrient. So in a lot of farming systems across Asia, we are finding substantial accumulation, so high concentrations of zinc and copper in particular, because of excessive use of chemicals.
Like
the other ones like boron and molybdenum, we really don't know. We don't know what the status is. So there's no set answer and there's no exact knowledge as to which one's best. It's more of a practical farm decision as to what works best for you. So if you've got drip irrigation, then fertigation through the drip is probably the best way. But if you're in a Sajan system where you can't do that, then foliar spray would then be the best. So it really depends on the farming system a lot.
Okay, thank you. Do you mean that manganese and zinc in Indonesia mostly have accumulated due to the excessive use of some chemicals like fungicide?
We don't know. I don't think we have the data for Indonesia, but I think in the limited samples we've done, we wouldn't see deficiencies of those as a problem.
Okay.
We think that the levels are relatively high. We don't know about upland soils. The new project will be surveying upland soils to identify what the limitations actually are in the upland areas. So we really don't know the status at this point in time. Yeah. The other thing that is complicated with some of the trace elements like boron are extremely light. So the atomic number for boron is 5. So it's a very low molecular weight. So volatility is very, very strong. So you will find that in areas where you have a lot of rainfall off the ocean, you can have substantial inputs of boron from rain. So in a dry climate like where I am, where we don't get a lot of rain, we could get boron deficiency. But in a wet climate where you get a lot of rain off the coast, you might not need it at all. It's probably there. The other thing, say, for example, in our Australian context, and where I am, our pH over time goes from neutral to 8.4 because of the irrigation water. Okay, so as we effectively lime the soil to bring the pH up, the boron, zinc, manganese all become less available. So you can't necessarily, in Australia, I'm not going to talk about Indonesia because I don't know, but in Australia, in my area, you can't think of the soil as being static. It's not stationary. It is dynamic and changing over time, depending on your irrigation inputs, your fertilizer inputs, your cultivation, a whole range of things that can affect trace element availability.
Okay. Okay, that's good. That's what is now the other questions from the chart.
Oh, why didn't I put fungi in there? Maybe because they're often very obvious when you've got fungal infections. Okay. Someone asked the question about fungal infections.
Yes.
They're usually, because they produce hyphae or spores, you can usually pick them quite easily compared with the other ones.
Yeah.
With the exception, I did mention verticillium wilt and phosphorus. So verticillium wilt is one where you don't necessarily see sporulation.
Yeah. Okay. It's nice. And then maybe any other question from not in chat but directly to address to Dr. Harper? Any audiences have any questions?
Oh, okay. Now there's one about nematodes.
Nematodes, maybe. Okay.
Yeah.
Maybe nematodes sometimes it's similar with nutrient disorders. Okay.
Look, what I would say, anything that affects the root system will affect nutrient uptake. So someone mentioned about fungi, soil pathogens, nematodes, bacteria, anything that affects the root system will affect nutrient uptake. So once you've got root damage, you don't get nutrient uptake. So they are related in that sense, yes.
Yeah. Okay. Okay. Are there any questions, audiences? And okay, so because this lecture is attended by multiple, I mean, mostly from plant protection.
Yeah.
And maybe they just have one subject in soil sciences, just basic soil sciences. Maybe Dr. you would like to explain some about what is the like mobile, immobile nutrition, and also the murder charts and what is the problem in nutritional plant disorders. So like murder charts, do you know
Sure.
murder charts, and then
Model.
about murder, yes, and then mobile and immobile. It's one of the strategy how to diagnostic which kind of nutrients disorder happen over there. Okay.
Which nutrients are mobile in the soil?
Yes,
you mean?
in the soil. Yeah, I mean mobile or immobile in plant and or in soil. I don't.
Yeah, two separate things. So the soil one,
yes,
mostly anions are mobile as, sorry, not quite right. Nitrate, chloride, borate would be mobile in a soil, would move,
yes,
because soils have negative charge.
yes.
So cations are held, anions generally move, but the exceptions are phosphorus and probably molybdate. So on acidic soils, acidic red soils, fixation of phosphate prevents movement. Although it still can move, if you put enough on, it'll still move. Whereas, of course, for cations, they're held on the exchange, but can still move. But because of the negative charge on soils, cations are generally held.
Yeah.
Within the plant, our knowledge of mobility of nutrients in plants has changed a lot. Mostly we would say the most immobile elements are copper, boron, calcium, but there is evidence of boron mobility now in plant tissue. The macronutrients are, with the exception of calcium, are mobile in the plant. And that's why they express on the older leaves, not the younger leaves. So they'll show on the older leaves because nutrients are moved out of the old leaf into the new growth. And most of the micronutrients, with the exception of molybdenum, are immobile and will mostly express on the youngest growth when there's severe deficiency.
Okay. Any other question from the audience? Please raise your hand or just address directly to Dr. Harper. Even Ibu Asti, any question or any comments?
Maaf Pak, izin bertanya ke Dr. Harper.
Okay. Just let's see in Indonesia, let's say in Indonesia also, or in English. Please try in English if you can.
I'll try, sir.
Okay.
Right.
Please address directly to Dr. Harper.
Okay, this question for Dr. Harper. Hello, Doctor. Thank you very much for sharing to us your experience and your knowledge about diagnostic pathogen or any disorder in the field. Thank you. So I can know specific difference maybe about the deficiency of micronutrients or macronutrients. But maybe I missed the specific symptoms of the biotic or pathogens. And maybe I intend to search your publication. Are all the diagnostic you experience already published in journal or paper, so I can search and study better? Maybe that's my question. Thank you very much.
Okay.
That's a good question. No, they are not published, not because I don't want to, but things like this are quite difficult to publish because they're not really a journal paper. They're just images that I've assembled over time. So they're not published. There are a lot of images in the literature, but with nutrition, you have to be careful to make sure whoever's authored them actually got it right. So you will often find in the literature where images are published as being a certain deficiency, they're actually wrong. The images are wrong. They're not actually what the author has said they are. So you do have to be careful. I would suggest that you read and look at images, and there's a lot of really good textbooks and farm notes and that you could access. I should publish them, but I don't know how I would publish all of my images, but it would be a good idea. In relation to micronutrients, it's probably important for you to understand that you don't necessarily have them as a problem in Indonesia. You might, but you might not. It might be more likely that they're not a major problem because Indonesia is a very young series of islands, and so the soils are extremely young. So when you have young soils, you may well find that these are not major problems for you. Our soils in Australia are ancient soils, very old and highly weathered. So for us, we may well experience these problems a lot more than you. So it comes back to understand your landscape, your climate, your crops is really, really important because they might not be a problem. You know, they might not be. Yeah.
Okay, thank you. Any other question? We still have for time, Wasti, until 10, maybe it's just 3 minutes.
Good time.
Okay, until 10 on Indonesian time.
Right.
So, yeah. If there are
It's lunchtime for Stephen.
nothing for you, it's lunchtime for you, but we haven't. We haven't. Okay. So if there's no any questions, I just would like to make highlights of this lecture. The accurate diagnostic is a must in what is in solving the problem plant health problem, and then the good diagnosticians must know any other other knowledge. I mean other disciplines like climates, soil, and many things about that, and also in agronomic and physiology, plant physiology. The good diagnostician have have the broad mind or broad I mean the broad mind, and then have broad knowledge about the others, because we are plant protectors mostly know that bacteria, virus, fungal, and something like that. But we sometimes ignore about the other, because other factors like nutrition also have a great effect to the development of the disease. So I think this morning is have broadened our knowledge about the plant health problem, because we have listened and hearing the what is outstanding lecture from Dr. Stephen Harper. Thank you very much for sharing your knowledge and practicing in Indonesia also. Stay healthy and stay safe during the pandemic. We wish you all the best health. Pardon?
Can I just make two comments?
Yes.
First one is for a field agronomist, because I've ended up being a field agronomist as well as a soil nutrition, whatever. You do need to draw on the experience of your pathologists and your and soil science, all sorts of people. Everybody has their knowledge, so it's important to make sure you draw on other people's knowledge. The second one that I would say is that experience. I've worked for more than 30 years in agriculture, and I always say, there's only one way you get experience, and that's to get hair that's my color. Right? And you can't buy it off the shop. You can't walk into the shop and buy experience. You only get it one way. So, you accumulate knowledge over time, and drawing on people's knowledge is a really important part of that. So, yeah.
Exactly. Agreed, Dr. Stephen. Okay, and then because time is up, and I hand it over to Ibu Asti. All right. Thank you,
Okay.
and thank you, Stephen Harper, for your sharing.
Sama-sama.
Okay, thanks, Pak Widodo. So at the end of this lecture, on behalf of the head of the department, we would like to present a certificate for Dr. Stephen Harper.
Thank you.
Yeah, our appreciation to Pak Stephen, for a very good lecture and sharing your field experiences. We hope that when pandemic is over, you can visit us again in Bogor. And we can go to the field together, and the students can experience by themselves about the plant health. So thanks again, Stephen.
My pleasure.
And also, we would like to thank you, Pak Widodo. I think we really appreciate your role as a moderator for this guest lecture. And finally, thank you very much for all the participants, the students in the Department of Plant Protection, the faculty members. I saw a lot of dosen here, Bapak dan Ibu. Terima kasih untuk partisipasinya. And please stay tuned, because we still have some more guest lectures coming this semester, and hope everybody can enjoy this guest lecture program from our department. So I think we finished this guest lecture session, and terima kasih sekali lagi. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Harper. See you in Indonesia after pandemic.
Thank you very much.
That's a good idea.
Thank you, Mr. Harper.
Thank you, Mr. Harper.
I'll contact you later, Asti.
Pardon?
Yeah.
Yeah.
Bye-bye.
Thank you, Mr. Harper.
Bye-bye. My pleasure. Talk to you later.
See you.
Thank you.